So thank you very much, everybody, for coming. We're delighted that you're here, and we're delighted to offer you some intriguing thoughts and discussion on an important issue, not only for technology, but also for the environment. My name is Oswald Schmitz, Oz for short. I'm the director of the Yale Institute for Biospheric Studies, YIBS for short. YIBS is sort of an umbrella institution here at Yale, incentivizing interdisciplinary or at least cross-disciplinary research and training in the environmental sciences. One of the nice jobs of YIBS, because it's basically, people liken it to a giant foundation within Yale. I get to give out a lot of money to incentivize things or do special projects and things. And this is certainly one of those. What we like to do in YIBS is occasionally have gatherings like the one we're having tonight and tomorrow to really discuss issues of importance and significance. And it's nice to be able to incentivize that and bring people from outside and across campus to sort of meet and discuss. And ultimately really try and find some resolutions to some of the reigning environmental problems. And that's one of the exciting parts of the job. This is more broad than the purview of YIBS because it involves, humanist perspectives and everything. So this is not just sponsored by YIBS, it's also sponsored by the Interdisciplinary Center for Bioethics at Yale and also the Franke Center for Humanities and Science. And it's a nice partnership, and it's also, again, an example of how we can gather resources at Yale to really sort of think about the broader picture and the bigger issues. I'm not going to say much more because this is not my show. This is really a show by a young woman who has had a lot of energy and passion to really wrestle with this issue. And she and I have been meeting almost weekly since the fall to really talk about this and get this off the ground. And so there's a lot of planning in here and a lot of hard work and a lot of passion, and that passion is Natalie Koeffler, Dr. Natalie Koeffler, who is currently in the Yale Medical School. And with that, Natalie, the floor is yours.
Can you hear me? Oh, maybe not. Hi. Thank you, everyone, for being here and accepting our invitation to join us in this very important conversation. I actually struggled to figure out what I wanted to say when I was going to have these short welcome remarks. I have a lot in my head and in my heart that I've been thinking about with this issue. It's a really interesting issue, and so couldn't really figure out what I wanted to say, and then I realized finally last night that really, this isn't about me getting up here and talking about gene editing to you and what I think about it. This is me getting up here and creating a space for us. And that's really what this vision for this whole summit has been, is creating space, opening space. And with that, we wanted to create a space where we can bring together people from all backgrounds and disciplines to be able to talk about this really important and complex issue, because this is a really complex issue. It's complex at the genetic level, where you have genetic networks that are interacting with each other, and we're trying to change those genes. It's complex at the ecological level, where we might be releasing organisms out into the environment that have mutations that we've initiated. And it's complex at the societal level, because there's these value systems that need to be tapped into. There's people's ideas of what's right and what's wrong that vary. And it also brings up these questions of really, what is it to be human? How do we fit into this greater space of our Earth? What does it mean? What is natural? What is unnatural? So these are really complex things and complex issues, and they require really complex solutions. Not complicated solutions, but complex solutions. Solutions that engage everyone and bring all the inputs and components together to be able to really start to figure out how to grapple with this issue. And that's what we're trying to create, a space where everyone can come together from their different backgrounds and join and discuss this. And that also requires a level of complexity on each of us as an individual. And what we invite you to do and what's important is that you come into the space in your entirety. in your totality and everything that makes you. And that's something I struggled with a lot as a scientist. I felt like as a scientist I had to leave certain things at the door. I had to leave my spirituality, I had to leave my values, my subjectivity. That didn't really help me in being as good of a scientist as I could have been. And that's something I think we all need to know, is to bring our whole being, our whole selves to this story. And that's our wisdom. That's our individual wisdom and knowingness. And from that individual wisdom, we can create collective wisdom. And I think that collective wisdom is what is so important in this issue. We find that space, that magical space where all of us can come together and try to steer this technology responsibly and in a way that really comes up with effective solutions that move us into an elevated future. And so that's what we're trying to do here, and I'm really so grateful that all of you have come to join in this conversation. I'm also so grateful for having had the opportunity to work with Oz and YIBS to put this together, for the financial support of YIBS, as well as Maj mentioned the Yale Interdisciplinary Science Center for Bioethics with Stephen Latham, who's one of our steering committee members there, as well as the Frankie program in humanities and sciences. We've also had great input from Misha Semenov, who's also on our steering committee. He's a dual degree major graduate student in School of Architecture and the School of Forestry and Environmental Studies, as well as Vivian Vigliotti, who is at the back there. She's been our communications and web design guru. Without her, you wouldn't have known that this is happening, so that was very important. As well as all the volunteers that have come and helped today and throughout, and people that just help shape the vision for this summit. This has been definitely very much a collective effort. I also want to give a very big thank you to the Yale Peabody Museum. They've allowed us to use this space for this event now, as well as for the cocktail hour, which will follow in the Great Hall. And Jonathan Rohner in particular has been really instrumental in helping with logistics of getting this event off the ground. And well, that's about it. I wanted to talk about just generally the program, so we'll have the keynote talks tonight, and then tomorrow, again, we'll be reconvening at 9 a.m. for breakfast, talks starting at 9:30 at Rosenfeld Hall, where we'll have a series of short talks ranging from geneticists to ethicists to architects to synthetic biologists to regulatory policy experts. It's going to be a really great discussion again tomorrow. So I hope you can join us again there. And now I would like to introduce our two keynote speakers for this evening. And I'm really just loving that the two of them are going to be up here to speak with you, because they really complement each other in a really interesting and wonderful way, I think. Our first talk will be given by Dr. James Collins, who's come from Arizona State University. He's the, let's see, I'm sorry, I always forget the full name of this. Well, he just basically is a professor of natural history and environmental studies at ASU. But he actually studies host pathogen interactions and with a specific focus on amphibians and how that directs populations in ecology. He's also was the director of population biology and physiological ecology program at the NSF from 85 to 86, as well as the NSF's assistant director of biological sciences. And he's really also helped contribute to shaping the space of ecological ethics and really looking, again, outside and broader than just the sciences to start communications about how we do science and how we look at ecology. He will be giving a talk called Altering Nature by Gene Editing. We can, but should we? And then Emma Maris will follow. Emma Maris is an author and writer who's written for a variety of news outlets, including the New York Times and Nature, Wired. And she also has written a really wonderful book which came out in 2011 called Rambunctious Garden, which really started to kind of, for me, reshape the way that we could approach environmentalism. And I think she will kind of touch upon these same kind of ideas of how we interact with nature and how we might need to change those interactions to make for a sustainable future. So without further ado, we'll bring up James for the first talk. Enjoy.
All right, well, it's a pleasure to be here. Natalie, thanks so much for that terrific introduction. I appreciate it. We'll do it. Great. So let me get us started here with three numbers. 438,000, 70%, and 2 minutes. 438,000 people, 70% of them are children, and 2 minutes. According to Target Malaria, in 2015, 438,000 people died from malaria. Now that number varies a little bit as I've looked at it, from 350,000 to the 438,000. 70% of those deaths were in children. And that figures out to about every 2 minutes a child dies as a result of malaria. Now, I want to give you a question with this information in the back of your head, actually in the front of your head right now. And here's the first question. This is not an exam, but you can think it through. So, if I gave you a technology that could eradicate a species of mosquito that caused that sort of impact as far as human populations are concerned, what would you do? There are your three choices in front of you right now. Now, my thesis that I want to explore with you this afternoon, explore with us this afternoon, is that there are genetic technologies being developed that can, in fact, reach into this space, that can help mitigate, potentially, that sort of impact as far as human populations are concerned. But like a number of technologies, and in fact technologies in general, as genetic technologies in particular have been developed, when you get into this sort of space, it begins to put a very sharp edge on a number of things. And in particular, the ethical, legal, and social implications of this sort of work, the sort of research that can enable us to reach into that sort of situation. And so I want to walk us through and help think about what this looks like today and how we might eventually think about this sort of problem. So to put this in perspective, the sort of genetic technologies which we hear so much about today are new and they're shiny and they're bright and they're just absolutely terrific. But in her relatively recent book, Helen Curry tells us the story of biotechnology, or tells us a story of biotechnology, and emphasizes for us the fact that thinking relative to manipulating genes, or at least manipulating inheritance, goes way back in the depths of the 20th century, the first decade of the 20th century, as a matter of fact. There were thoughts about how might you manipulate genetic material using things such as radiation, using a chemical called colchicine to actually change the information in organisms. And then she walks us through this history, reminding us that there's an arc of change associated with what we now know as biotechnology that stretches from the first decade of the 20th century up until the present. So what I'll talk about today, and what in fact we'll talk about for the next 2.5 days, is part of an evolving story. It's part of a story as far as discovery is concerned. It's part of a story in terms of application of that discovery, in terms of biotechnology. And it's a part of a debate that's gone on now for the better part of 120 years in terms of how we can take this information, use it, and move on. Now, the thing that's happening, though, today is that these technologies, especially the ones that I'm going to talk about, have become so precise and in some ways so easy to use that they really begin to put a sharp edge on this conversation between scientists, ethicists, legal scholars, social scientists, causing us the need to think about these problems as multidisciplinary, transdisciplinary, solutions that are probably going to be integrative. They have to be integrative. And the question is how to move ahead in that sort of space. So, on the same website of Project Malaria, we see Target Malaria, sorry. They proposed a possible solution in October of 2016. Could gene drives, would it be possible to use gene drives to tackle malaria in Africa? This was an intriguing possibility, and still remains an intriguing possibility, as far as a way to reach into these systems and have an immediate and interesting effect. So let's talk about what a gene drive is in order to get our footing there. And I'm going to walk us through two cases, one involving gene drives very directly, the next one involving a related form of genetic modification. And then we'll get to the point of, all right, how might we make some decisions in this space? Or what would you need to know? What would you want to be thinking about in order to make decisions in this sort of space? So here's what a gene drive is. I won't read it for you, but the point that's relevant is this notion of guaranteeing transmission of a gene from one generation to the next. So let's talk a little bit about that, what that means. So in this audience, we have men and women. The ratio of men to women across the population, our population at large, is 50-50, 50% males, 50% females. At birth, on average, there are as many boys as there are girls. So we talk about the gene, let's say the gene for maleness, being transmitted with a frequency equal to the gene for femaleness. And that's because males are typically XY. Chromosome 1 is XY, chromosome is X, chromosome 2 is Y. They're paired. And then for women, it's XX. And so when you mate, you wind up with a 50-50 sex ratio that falls out of that. Now, the notion of guaranteeing transmission to the next generation would be a situation in which, for example, males make only Y-bearing sperm. Only Y-bearing sperm. And if women continue to make only X's, that means with each reproductive event, you get only males. XY, 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 only males from one generation to the next, if it were the case that you could have every male make only Y sperm, Y-bearing sperm. The technology I'm going to talk to you about now, this notion of a gene drive, can do just that, or at least in principle can do just that. And it rests on this bit of molecular biology, or it rests partially on this. It's a term, or at least a word, an acronym you may have heard. When you go to your next cocktail party, you can tell them about clustered, regularly interspersed, short palindromic repeats, and they really be intrigued or ask you to leave. One of the outcomes is going to be the possibility. But the imagery that goes along with this notion here is, first off, the imagery associated with a scissor that can cut DNA. And the other sort of imagery that's used in connection with CRISPR is the notion of a word processor. Cutting, copying, and pasting new information into the genome. Cutting, copying, and pasting. Let's see how that might work. So let's say we have a mouse. It's got a pair of chromosomes. We're only going to have 2 chromosomes in this mouse. That makes it brown. And we'd like that mouse to be green. So we construct a bit of DNA. That bit of DNA has 3 components to it. It's got the change that you want, so we're going to cause this mouse to be a little bit green. We're going to need something to guide our new information to where we want it to be in the chromosome of this mouse. And we're going to have this CRISPR-Cas9. I won't go into CRISPR. We have the big wording up here. That's the part that does the cutting. So here's what happens. You have a scissor. You need a guide. Make a space. move the new information over. And our mouse is now green. We've introduced the genetic material to turn that brown, so when that brown mouse begins to breed, it has green offspring. Furthermore, because we still have the cutter in the system, the one chromosome reaches over and cuts the other chromosome and replicates that fraction of the genome into it. So now we have a mouse that's green and it has 2 chromosomes carrying the same information. Make a green mouse. We're going to breed that green mouse with another brown mouse. Cutting happens, moving happens, and you wind up then with now green mice. Furthermore, the green mice have the same chromosome. They're bearing the same genetic material now. And this goes on from generation to generation until by the 5th generation, we have only green mice. Only green mice. Now, if that bit of genetic material that I moved over said, make the developing embryo a male, then by the time I get to the 5th generation, all my mice are males. Not green. In this case, they're all males. And if they're all males, and it's a sexually reproducing species, it's a population doomed to extinction. And if that trait moves out of that population and into other populations, you have what's called a global gene drive, which means you could drive a species to extinction. Okay? That's the technology. This technology has been developed now and has been demonstrated in the laboratory in Drosophila, small fly. It's been demonstrated in a species of yeast, and it's also been demonstrated in 2 species of mosquitoes. All in the laboratory, not in the field. A couple of facts about gene drives that are worth keeping in mind. They occur in many species. The reality of driving genes has been known for the better part of 100 years. The difference now is having the technology to be able to harness those natural biological processes towards the sort of ends that I'm suggesting. Gene drives are designed to spread and persist. So unlike a manipulation that Monsanto might do, where it wants to keep it in a particular farm or cluster of farms, this is designed to spread. And it will move beyond boundaries, other than those in the laboratory. Sexual reproduction is required and a relatively short generation time. So we're not going to use gene drives in humans. There's no point. 20-year generation time or shorter in some societies. It's not what you want. You want it to be able to turn quickly in these populations in order to convert them, as I said, from green mice or from males and females to all males. I had a chance to look back at Silent Spring, and indeed, here's the language that Rachel Carson used for biological control. So a gene drive is a kind of biological control. It is, in the way that she could so elegantly put, it's the notion of turning the strength of a species against itself. It's the idea to use the drive of an insect's life force, to destroy it. It's just this elegant way of putting what this biology is all about. Now, the thinking in gene drives, on the upside, in terms of this technology, is you can apply it in a variety of different ways. Think about it, as I suggested early on, to be able to manipulate populations of organisms that are bearing infectious diseases. Think about it as being able to remove exotic species from island populations where endangered species of birds are threatened. Think about it in terms of being able to reverse herbicide resistance in agricultural situations and make those plants once again susceptible to herbicides. So these are the contexts in which it's being thought about to use these gene drives, and in fact, it has attracted a lot of attention. The New York Times had a story, has had multiple stories actually, on this technology, and the use of the notion of using it against infectious diseases in our species and in crop species. It has attracted the attention of conservation biologists. So in this article by Michael Spector, he's had a couple of articles now in the New Yorker, he's arguing, and conservation biologists, some are agreeing with him, to use gene drives to remove mosquitoes from Hawaii, where they're an introduced species anyway, and they're driving the extinction of native Hawaiian birds. In fact, the evidence suggests they've already driven a number of species extinct. It's actually, in his words, exciting information for conservation biologists. And in fact, in a recent report, there's a series of workshops that are happening to think about the possibility of driving mosquitoes out of Hawaii. It's an exotic species, and this is a way forward. And even in the scientific community, we have a leading individual endorsing the prospect of driving these malaria-bearing mosquitoes, or at least one species of them, to extinction. It's also been picked up in the popular press in some ways we might describe as a little bit more lurid, or you do begin to get on a slippery slope anyway. If it's mosquitoes today, all the mosquitoes today, I bet I would get a positive vote in Connecticut if it were the ticks that are bearing Lyme disease tomorrow, for example. But where do you stop once you're on that slope? And who gets to make those decisions? And where do we have those conversations? Finally, you can get one step even more lurid, if that were possible, and begin to talk about an extinction machine here in terms of the MIT Biotech Review, classifying this as an extinction machine. So here we have a case where we've got this genetic technology. We want to reach into natural systems, and Emma's going to help us understand what natural means. We're going to reach into these systems and initiate these changes. So that's question one. So you can go to your cocktail parties. Remember, you're going to talk about CRISPR, and you're going to talk about, should I use this technology? Here's question two. Let's say I gave you a technology capable of bringing species back from extinction. So we're not driving them extinct now. We're going to bring them back. And what would you do? 3 choices. It's an open book exam. So some examples here would include the American chestnut. So the exact definition of de-extinction moves around a bit. So for some, this is an example of de-extinction. For others, it's an example of genetic rescue. And in fact, in a moment, I'll show you the two of them tied together. So the American chestnut, you can still find individuals, but they're not the canopy trees that they were in this northeastern deciduous forest in which we sit now. They're basically remnants of themselves. And there are a couple of different ways in which genetic modification is being tried in these organisms. One is through transgenics. So William Powell, who's over in SUNY Syracuse, is using transgenics. There are other groups that are breeding American chestnuts to Chinese chestnuts, where the fungus originally evolved, the blight originally evolved, and are trying to breed resistance into lines of American chestnuts. So that's one way to do it. But the other way to do it, that's being tried in other species as well. So in the middle, we have this lovely black-footed ferret, and they are down to a small number of individuals. But there, again, are research labs thinking about putting into these populations the genetic capacity to resist the infections that are killing these populations. But on either side, left and right, we have even more iconic examples of the notion of de-extinction. On one side is the passenger pigeon, where there's a program in place now to try to bring back passenger pigeons using the DNA of individuals that are present in, for example, museum collections, such as this museum here. And or the notion of bringing back woolly mammoths. Now, this can be taken to the point where some of you may have seen in the Atlantic a little while ago, the notion of Pleistocene Park, where you can bring back, or the thinking is to bring back some of these organisms and have them once again present on the steppes, the high Arctic, where they once existed. There are a number of individuals that are thinking very hard about this, thinking hard about it in very interesting ways. Dr. Beth Shapiro, for example, is being very clear about how hard it is, in fact, impossible probably to bring back a mammoth as such. You can get bits of mammoth DNA out of some of the historical record, but you can't get the whole genome. But you could put bits of mammoth DNA into elephant DNA, and in particular Asian elephant DNA, which is what George Church and his lab are doing at Harvard University. So you can begin to get contemporary organisms with traits comparable to woolly mammoths that might have a better chance of surviving in the high Arctic. And Stewart Brand, who has thought a lot about this notion of de-extinction, has made this point here, which really does begin to push this notion of technology, again, up against the ethical, legal, and social implications of this sort of work, and raises the prospect of this being a moral obligation on the part of contemporary society to do something in terms of bringing these organisms back. And Michael Archer, has made an even stronger case that if we're responsible, he argues that we have a moral obligation to try to bring back such a species. Interface of ethical, legal, social, science, humanism. How did these things come together in order to make a decision in moving ahead? So the other thing to keep in mind for your cocktail party is that when you hear the word extinction, I'd love for you to think, I wonder what sort of extinction this individual is thinking about. 99+% of the species that have ever been on the planet have gone extinct. We can call that natural extinction if you want. There are these genetic technologies being developed that have the capacity to do deliberate extinction, and then there's the notion of bringing organisms back, de-extinction. So extinction can mean a variety of different things and can play itself out in a variety of different ways. So let me now go to this third part of the talk and talk about, okay, we can, but should we? And how do you think about this kind of problem? How might we think about it? Now, in the United States, when you have a complex problem like that, one place you can go is to the National Academy of Sciences. And indeed, the National Institutes of Health and the Foundation for the National Institutes of Health came to the National Academy in 2014 and said, we'd really like to know some things about gene drives. Could you put together a committee for us to take a look at it? I co-chair the Board on Life Sciences for the Academy, and they asked me to chair the committee. I co-chaired the committee for them. I brought in Elizabeth Heitman, who is deeply trained in ethics, and the two of us co-chaired the committee for the Academy of Sciences. And what the NIH asked us to think about were these 4 questions. What might be the unintended consequences of something like gene drives? Do we know enough to use them? Should they be used, and even should they be developed? And how do we decide these sorts of things? So we took a look at the science. And in looking at the science, we decided that the science was not sufficiently well developed to be able to recommend releasing gene drives into the environment. That we did not know enough about the ecology, ecosystems biology, the evolution of these systems to be able to make the kinds of predictions needed to develop the models yet to be able to justify releasing these gene drives. But we did argue that the research should continue. The benefits of this sort of research are just so significant. The notion of being able to lift the disease burden on some populations of humans is just so significant that it made a lot of sense to be able to go ahead with this work. But we also recommended that if this work is to go ahead, it had to be firmly grounded on values. Values, as we looked at it here. Complicated, evolving beliefs in our lives, in the world at large. And these values had to be, this notion of values that had to, things that had to be protected, fostered, and indeed even avoided. These values had to be coupled with the idea of engaging the public. And engaging the public at different levels. So if you're going to eliminate mosquitoes in Greenwich, Connecticut, you want to talk to the people in Greenwich. But you might also, in fact, you'd really want to talk to the people here too, especially if you're going to do a gene drive. And maybe even the people in New York. And then you can go to even larger publics that might have a stake in thinking about whether or not this technology should be developed and then used. We also thought a lot about governance. And the idea that these gene drives are presenting important and interesting environmental and security challenges as well as benefits. So governance of these gene drives was going to be important. And furthermore, we were also thinking about the notion of anticipatory governance. The idea of thinking hard about a technology and its implications before it's fully developed and ready to be released. You really want to have thought about it as deeply as you can beforehand in anticipation of releasing it. And we saw this as a way to help mitigate risk. We saw this as a way to be precautionary. And we argued that there were ways to be precautious but also consistent with supporting science. So it was not the precautionary principle in the sense that you just don't move ahead if you don't know where to go. But rather, this has to be an active process of deciding where you might want to go, how you might want to get there, can you get there, what are the issues that need to be dealt with in thinking this through. And the imagery that Science magazine used for us, for this particular article anyway, was the notion of left-hand side, as you're looking at it, places not to go, right-hand side, blinking yellow lights, be cautious, but we could think about how to get there. Now, if I'm going to make an argument consistent with a good rhetorical argument, I'm going to give you the counter-argument. And the counter-argument is that just don't go there. And these two distinguished individuals in fall of 2016 at the International Meeting for the Union of Conservation of Science, Conservation of Nature, made the case that you shouldn't go there in terms of gene drives. It's just too risky. We don't know enough. And this sort of technology is not something that we want to use in the natural world. In a larger sense, there's a big argument about the degree to which we want to be reaching into manipulating natural systems. If technology helps us save the wilderness, will the wilderness still be wild? There is a very definite view that there are parts of the world we just don't want to be touching in terms of the use of these technologies. And what gets to count as those parts of the world is a big discussion in and of itself. So to wrap it up, you see this interface between scientific freedom and responsibility. The freedom to move ahead on the part of scientists to develop the kinds of things that can be immense benefits as far as society is concerned. But you wind up needing to deal with immense issues related to responsibility. You have the notion of driving a species extinct, mosquitoes, let's say, traded off against using that as a way to mitigate an immense burden of disease as far as human populations are concerned. Where do you strike that balance? And how do we decide how to strike that balance? And in a larger sense, is this our view of nature in terms of a place where we visit, leave the fewest footprints possible, and then come away from it? Or do we have a view of nature that is much more complex? A view of nature that comes to us in pieces, that is really a jigsaw puzzle for us to put together, solve. There are pieces still missing, piece that we can think about filling in, and how we fill it in, and how we do it, and what our view of nature is going to affect all those kinds of things as we go forward from here. So my last thought would be something along the following lines. If you go to Washington, D.C., and you go to the far northern part of Georgetown, you'll find the library there, in which you can walk in and see chiseled on the side of Dunbarton Oaks its mission, or part of its mission. And part of its mission goes to clarify an ever-changing present and inform the future with its wisdom. So it seems to me, as we think about these complexities and the interface between these sorts of genetic mechanisms and our ecosystems, we can do not much better than thinking along those lines. Clarify this ever-changing present and use our information, our vision, to inform the future for our children and our grandchildren with just as much wisdom as we can emerging from this sort of work. Thanks very much.